Friends, welcome to Biology Made Easy. We are discussing the different non-allelic gene interactions where we have discussed the complementary genes, supplementary genes, epistasis, duplicate genes and polymeric genes. So in this series, today in this video we will discuss the last non-allelic gene interaction, that is the collaborator genes, or we can tell it as modifying genes, which gives rise the same ratio, that is 9:3:3:1. So what is this collaborator or modifying genes? There are two non-allelic genes which have their own effect when they are present in the dominant form and individually. But when both of them combine together, they give rise to a new trait. For example, one dominant gene is A. So when it is present, it will give rise to a character, suppose that is X. There is another non-allelic gene, that is B, and this will give a effect which is Y. So when individually they are present, they are giving a particular type phenotype. But when A and B will combine together in one organism, it will give rise to a different phenotype, that is Z. So here you see what is the difference between the polymeric gene interaction and this collaborator genes. In case of polymeric, both A and B were giving the same character, but here they give rise to separate characters. But when they combine together, they give rise to a new trait. And when they are present in a recessive form, that means small a, small a, small b, then that will also give another type of character. That is why we will get this 9:3:3:1. So what is the, again I am repeating the definition. There are two non-allelic genes. There are two non-allelic genes which produce their own phenotypic effect when present in a dominant form and individually. But when present together, they give rise to a new trait, as we have discussed here. So the best example to explain this collaborator genes is given by Bateson and Punnett in case of the shape of the comb in poultry or chicken. So if we take that example given by Bateson and Punnett, they conducted certain experiments in case of the comb types. Bateson and Punnett, they conducted some experiments in the comb shape of poultry, or we can call chickens. So in this case, what they found when the gene capital R is there, it gives rise to rose type of comb. And when it is present capital P is there, it gives rise to P type of comb. And when both capital R and capital P are present, it gives rise to a new type of comb, that is known as walnut comb. All in the case of a poultry. And when they are all present in the lower case, small r, small p, then it gives rise to leghorn comb. That means there are 4 different types of comb structures in case of poultry. Now, if a cross is made between two types, there are different types of poultry having different comb types. So, Peterson and Ponnet selected two poultry varieties. One is Wandots with a rose comb. And their structure is capital R, and small p. The name of that variety of poultry is Wandots. So these were crossed with Brahmos with P comb, and their genotype is small r, capital P. Now, when the cross is made, the F1 hybrid will be capital R, capital P, small p. But the shape of this poultry is walnut. That means cross was made with a rose comb type with that of a P comb type. But the F1 hybrid become with a walnut type of comb. Now, when two such walnut types are intercrossed, capital R, capital P, small p, when they are intercrossed, what will happen? In the F2 generation, N will get capital R, capital P, capital R, small p, small r, capital P, small r, small p. And also in the female side, we'll get capital R, capital P, capital R, small p, small r, capital P, small r, small p. Now, if we draw the checkerboard, what are the different characters? Let us see. Now, in this case, you see, in this place, both capital R and capital P is there. Here, capital R, capital P and small p. Here, capital R, small r, capital P. Here, capital R, small r, capital P, small p. Here, capital R, capital P, small p. Then here, capital R, small p. Here, capital R, capital P, small p. And here, capital R, small p. Similarly, here, it will be capital R, capital P. Then capital R, capital P, small p. Then here, small r, capital P, small p. And here, small r, capital P, small p. And here, capital R, capital P, small p. Here, capital R, small p. And here, in this case, small r, capital P, small p. And here case, all the four are in their recessive type, lower case. Now, already we have written here, when both capital R and capital P are present, then the comb is walnut type. That means this is walnut type, this is walnut, because both the dominants are there. This is the walnut, and these are also coming under walnut. And again you see here both capital R, capital P is there, so this is also coming under walnut. Here both are present, so this is also coming under walnut. That means the walnut type comb, they are 9/16. Then you see at how many places only the dominant gene R is present. Only dominant gene is present in this case R. So naturally it will give rise to rose comb. So this will give rise to rose comb, this will give rise to rose comb, and this will give rise to rose comb. That means the rose comb type, it is 3 by 16. Then you see in how many cases only capital P is there. So only capital P is present in these 3 cases. So this will give rise to, it will give rise to P comp, this will give rise to P comp, and this will give rise to P comp. That means the P type of a comp, then this is also 3. Then the last one, in case of last one, all are in the recessive form. So this is the Leghorn comp. So the Leghorn comp, it is coming in the proportion of 1 by 16. So if we now analyze, that means we are getting all the 4 characters in the ratio of 9:3:3:1. So the F2 phenotypic ratio is 9:3:3:1. This is similar to that of the F2 phenotypic ratio of Mendel's dihybrid cross. But what is the difference you see? In this case, what happens? This is the Rose type. This is the parental type. This is P type. These are the parental types. That means these 2 are coming under the parental types. Parental types. But what about this? This is a completely new character. When both of them are present in dominant form, and this is also a completely new character. That means the middle 3, they are the original characters. But when both of them are combined together, they do not give rise to recombinants. They give rise to a new character. But what is happening in case of Mendel's dihybrid cross? In case of Mendel's dihybrid cross, this 9 and 1, these were the parental types, while these 2, they are recombinants. That means if this was suppose round with yellow seeds and this with wrinkled and green seeds, then in these 2, the characters are just reshuffling due to independent assortment. That means round was combining with the green and wrinkled was combining with the yellow color. That means same colors are only reshuffled, but this is completely different. That means one type of capital gene is giving rise to one character, the other is giving rise to one character, but when both of them combine together, they give rise to a completely new character. What is the reason behind it? The reason behind it is the biochemical reactions. That means for production of this walnut type, which requires 2 dominant genes, that means inside the organism there are certain biochemical changes. There are production of different chemicals due to enzyme production, which requires the presence of both the dominant genes so that a new character is produced. So that is why this is known as collaborator gene. That means both of them are collaborating with each other to give rise to a new trait. So this is completely different from that of the Mendel's dihybrid ratio. So this is the brief idea about the collaborator or modifying genes, where the ratio is the same, 9:3:3:1, just like that of the Mendel's dihybrid cross. But here the regions for production of this 9:3:3:1 ratio is different from that of the Mendel. So I hope now a very clear understanding about the collaborator genes. So thank you very much for watching the video. God bless you. Have a good day.